Oops. Okay, well, we might as well get started. So thank you to Dr. Trevor DeVries for agreeing to come and talk to our give our CNM seminar this month. So for anybody who doesn't know Trevor, Trevor's a professor and Canada Research Chair in Dairy Cattle Behavior and Welfare in the Department of Animal Biosciences. After completing his BSc and PhD at the University of British Columbia, he joined U of G as faculty in 2007. And for the first 8 years, he was located at Kemptville campus, and since then has been here with us at Guelph. So in his position, Trevor leads a really highly productive research program that's focused on dairy cattle behavior, nutrition, management, and welfare. So today, Trevor is going to talk to us about how good behavior leads to good nutrition in dairy cows. And so I will pass the floor over to Trevor, and thanks very much, Trevor.
Perfect. Thanks, Jen. You can hear me well?
Yes.
Excellent. Yeah, and thanks everyone for being here. I'm going to break the mold a little bit from the last number of CNM seminars. It's just going to be me presenting today, so if you were hoping to see my grad students present, then I'm sorry about that. A couple of reasons for that. One is a lot of my students, actually, I'm kind of in a transition right now, students that are kind of starting off in terms of their projects, and then the few few kind of ongoing students that I have right now are quite busy with their current projects, and I have one that's quite busy with preparing for a qualifying exam right now too. So, but I am going to be talking about some of their research, some past and kind of present and ongoing type stuff through this presentation. Kind of splitting it up into two areas. One is kind of why we're interested in this topic of behavior, and specifically feeding behavior of dairy cows. And then talk a little bit more specifically about some of our current projects related to behavior of cows and how that ties in again with nutrition and nutritional outcomes in dairy cows. And we'll hopefully give enough time at the end. We'll run through this and hopefully have some time for discussion after the presentation. I started a lot of presentations with this question about why we care about feeding behavior and of dairy cattle specifically, and my simple answer is that changes in intake are mediated through changes in feeding behavior. And when we're thinking of dairy cattle specifically, where a lot of my program's focus is, a lot of things are driven by intake, right? So production at the end of the day and health are going to be driven by nutrient intake and the amount of that. And really, at the end of the day, if we want a cow, say, to consume more feed, we need her to change some aspect of her feeding behavior. And so, and that's just mathematical, right? So dry matter intake is really a function of how many meals a cow has per day and the size of those meals. You multiply one by the other, you get the intake. Same thing goes with the amount of time an animal spends eating multiplied by the rate of consumption, you get intake. And so really for that intake level to change, we need some aspect of their eating behavior to change, or multiple aspects within that. And so from that perspective, we have tried to look at this question of what is kind of most important in terms of driving dry matter intake, particularly in, say, high production dairy cows. And one of the analyses that we did a couple of years ago shed some kind of interesting results on this. And this was actually started with an undergraduate project with Carly Johnson, who many of you know works in the OAC Dean's office now. When Carly was a fourth year and she did a research project class with me, I tasked her with taking a data set from 5 previous studies that we had conducted over the years, where we had intake, production data on cows in those studies, as well as detailed measures of feeding behavior. And what I got Carly to do was to do some exploration in how those behavioral measures were associated with intake. And then we ran some more in-depth multivariable models to, say, predict dry matter intake based on those various inputs, behavioral inputs, and production actually as well. You can look at the paper for that. But from an intake standpoint, what we observed was that the two most consistently associated variables with dry matter intake in these higher production cows were the amount of time cows spent at the feed bunk per day, as well as the number of meals cows had. What you see on the screen here in the data is the time associated with intake. And there's a couple of things to note. It's not a perfect relationship. We're only explaining about 25% of the variation in dry matter intake. And there's a whole bunch of other things going on there. And we see a huge spread there in terms of the amount of time, say, that cows are spending at the feed bunk per day eating their feed. But for those cows in general, what the data would suggest is that those cows that are consuming a lot of feed, they are, again, having to spend a significant proportion of the day there. And same thing with the number of meals they had per day, that was also positively associated with that intake, more so than, say, how fast the cows were eating or the size of their meals. And so, again, this data is a little bit, from a practical application standpoint, tells us a little bit about how we should be focusing kind of management and even the diets themselves in terms of promoting those types of eating patterns in cows. In addition to the time course of eating, the other kind of behavior and what we classify as a feeding behavior, which is important and hugely important for cattle and ruminants in general, is rumination behavior. And rumination obviously is what makes ruminants unique. Has various functions, including salivation and buffering associated with that. But probably the biggest thing is actually, breaking down feed particle size, which is then going to have an impact on digestion rate and potentially even passage rate. And so rumination behavior in itself, we know should be related to intake. Not only is it a function of the amount of feed they consume, so particularly the amount of fiber consumed is going to impact how much rumination an animal does. But within, say, a diet, we also know that, or within, say, given dietary parameters, we would assume that greater rumination would potentially increase the rate of digestion, passage rate, and then also, say, allow that cow a quicker return to eating, which is then going to potentially increase the amount of feed she can consume as well. And so cause and effect there, not always known, but at the end of the day, the data would again suggest that in general, we have a positive association there between rumination time and dry matter intake. Again, not a massively steep, as you can see here from just as the simple linear model here, but not a hugely steep fit line here. But again, those cows that are consuming the most amount of feed, at least within this data set here, are the ones that are spending the most amount of time ruminating and chewing their feed per day. And so from a practical application standpoint, again, that anything that may potentially limit their ability to spend time ruminating may have a negative impact on their ability to, say, maximize their dry matter intake. And that kind of raises another question, which is a more recent focus within our group, was whether or not it's important to consider when and where rumination occurs, say, for a dairy cow. We know that when is typically obviously, and this is some data from another group, but showing that, rumination typically occurs reciprocal to intake patterns. So if you look at a diurnal intake pattern of dairy cows throughout the day, so dry matter intake per hour, the rumination kind of maps reciprocally to that, right? So they're going to be, obviously, while they're eating, they're not ruminating, and then the rumination bouts follow those intake bouts in those cows. So we typically see more rumination in the middle of the day, and then the late evening and early morning hours. Those time periods of the day are also associated when cows are lying down. And there's been some hypotheses whether or not, say, rumination is more effective while cows are lying down versus any rumination that, say, may occur while cows are standing up. And some estimates from previous studies would suggest that cows spend, generally somewhere between maybe 60 to 70% of their total rumination time lying down, where the rest occurs while the cows are standing. And there's some question about whether or not, and there's variation around that between cows. And so the question is whether or not that kind of time distribution of lying versus standing and the rumination that occurs there influences, say, the outcomes of that behavior. And we did a little project last year to try to at least start to dig or kind of scratch the surface on that. And this is again, actually an undergraduate project that I worked with Clay McWilliams, who was a BSc AGR student last year and graduated this past spring, and has currently or since joined my lab as a master's student. But what I tasked Clay with was, again, as part of his fourth year undergrad project, was to take a data set actually from two previous studies that we had of cows that were kept in studies at Elora, whereby we had data on the rumination behavior of the cows, we had data on the standing and lying behavior of the cows, as well as the feeding behavior and feed intake and production of those cows. And what I asked Clay to do is put together that data to look at the associations between those behaviors and whether or not we could determine whether or not those cows that, say, spend more time ruminating while lying down versus standing up, whether or not there was associations of that or between that and, say, their intake and productivity. One of the challenges that we had in that data set is that the rumination collars that we use and many people use, we don't actually get minute-by-minute rumination time. We actually only get rumination time in a 2-hour time window. So we basically get the number of minutes per 2-hour time block per day whereby they're ruminating, whereby for measures of, say, standing and lying behavior that we measure with a data logger or feeding time that we get from the feed intake bins, we get it on a minute-by-minute basis. And so we could not actually line up that data perfectly on a minute-by-minute basis. And so we actually had to sum the data down into 2-hour time blocks. And so because of that, we couldn't say exactly when they're standing and lying and ruminating down to the precise minute. Because of that, we actually had to calculate a probability that they were, say, ruminating while lying. And so for every 2-hour time block of the day, what Clay came up with was this probability, which basically was the amount of time per 2-hour time block that they spent ruminating multiplied by the amount of lying time divided by that 2-hour time block within. So lying time within that 2-hour time block divided by 120 to basically get a probability that the rumination and lying time during that time period, that 2-hour window per day, was occurring at the same time. And then once we had those probabilities, then we use that probability both as an outcome variable as well as a predictor in some statistical models to look at the association of that probability with various things, including the total amount of, say, lying time and rumination time of those cows, which again, kind of not too surprisingly, but kind of proves the measure in itself, just showing that, yeah, those cows that are spending more time lying per day are also likely to be spending more time ruminating per day and then have a greater probability overall of doing those two things at the same time. And so again, that shouldn't be too surprising for us. But what was interesting is that we then used that probability as a predictor of the dry matter intake of the cows. And what we observed there was a tendency for an association whereby those cows with a higher probability of having rumination and lying behavior occur at the same time were tended to have higher dry matter intake on a per day basis. And this is kind of interesting. Again, there's been some hypotheses around this in the past, whether or not potentially rumination maybe is more effective while cows are, say, lying down, whether or not that maybe actually promotes greater rates of passage. And that might be the reason why we're seeing greater dry matter intake in these cows that spend a greater proportion or have a greater probability of doing more ruminating while lying down versus, say, standing up. Again, this is kind of a preliminary analysis and the first kind of crack at this, but it does open up the door for some more work to probably further explore that. Similarly, we also demonstrated an association of that probability actually with milk fat and protein content in that data set as well. Again, as you saw in the previous slide, there was in this case, it's a tendency with milk fat, and it was statistically significant in terms of the association with milk protein. Not perfect relationships by any stretch, explaining again about 20% of the variation in each of these outcomes. But definitely a positive association whereby we're seeing higher milk fat, higher protein content in those cows that have a higher probability of ruminating and lying at the same time. And again, some hypotheses that we threw around in this, the potential that maybe, and again, there's been previous work suggesting that maybe there's greater salivation and salivary flow in cows that ruminate while they're lying down, and that might then contribute to improved rumen environment, more stable rumen environment, and thus improvements in terms of overall digestion and digestion efficiency, and that might be contributing to these things. But again, preliminary stuff that needs to get further explored. We did have some rumen pH data from some of the cows within these studies, and we were hoping to look at that, but there just wasn't enough data there to really explore that well. And so that's something that we hope to do maybe in some follow-up studies to this in the future. Kind of on that vein then, we also know that the feeding behavior of cows specifically might be directly related to rumen function and health. And largely due to just the patterns of consumptions that we see in dairy cows. And this is kind of a simplistic view of this, but this is just an example of a pH trace, a rumen pH trace of a cow, whereby we can see that drops or declines in rumen pH, reflecting accumulating VFA in the rumen, would correspond to times of feeding activity, and specifically in this cow here, where we see 4 kind of drops below a pH of 6, those corresponded very well with larger meals that that cow consumed just prior to those drops, that whereby the highly fermentable carbohydrates are getting digested fairly quickly, and we see that accumulation of acid and that drop in pH that occurs. Now, the reason I'm saying all this is that we know that again, the larger and longer these drops are, say, within day, the more potential negative effects there may be on the rumen environment in terms of the activity of fiber digesting bacteria, and then resultant from that, say, the rate of digestion of fiber, which is really one of the limiting factors when it comes to intake and return to eating in cows. And then there can be other effects of that as well. We know that large drops in pH then also result in a decline in saturation of fatty acids within the rumen, and some of those fatty acid intermediaries, the unsaturated fatty acid intermediaries, may then also be absorbed and actually block milk fat production at the mammary gland. And that's the reason we see that association between, say, low rumen pH and ruminal acidosis and say milk fat depression in cows. And so then it makes sense that intake patterns resultant from, say, management may, say, influence some of these outcomes. And just to highlight that or to show that association, this is some new data that comes from our group as well, This was a study done by Marcelo Castro, who was a visiting PhD student with us last year. He was working on a data set that we had of, it was actually a herd level study, 124 robot farms in Quebec and Ontario, whereby we were looking at the bulk tank fatty acid profile on those farms. And one of the outcomes that Marcelo observed was the fact that herds that fed more often, so delivered their PMR, and these are robot herds, so the partial mixed ration at the feed bunk, that were delivering feed 3 or more times per day. Many of those farms feeding more than 3 times per day were feeding with, say, automated feeding systems. We saw a greater proportion of de novo fatty acids in the milk of those herds. And we know that the de novo fatty acid proportion in milk is reflective of the amount, obviously, of milk fat that is produced within the mammary gland, which is going to be a reflection to some degree of the not only available substrate there, but also potential, as I was mentioning before, maybe things that may potentially limit their production of these fatty acids in the mammary gland as well, and so would be reflective of, say, an improved rumen environment when we see a greater proportion of, say, those de novo fatty acids. And so really, long answer short is that, yeah, we know feeding management can influence these eating patterns, which then may influence rumen fermentation and then have outcomes affecting, say, milk composition. One other aspect of behavior, which again, our group has focused fairly considerably on in the last number of years, is what cows actually consume. So not only just the time course of eating, but the actual act of consumption, and specifically looking at how cows sort their diet. And in the dairy industry, we provide this total mixed ration typically, which is supposed to provide the balance of nutrients that dairy cows need relative to their requirements. The challenge there being that if the cow does not eat that diet as it's put in front of her, then we don't necessarily expect to meet our targets of, say, production and health outcomes, et cetera, that we would want from that diet. And again, that can be a challenge, and we're currently doing projects looking at ways of mitigating that behavior, because the reality is we don't see cows necessarily meet the targets that we expect from them based on this behavior often. And just a couple examples of that, a study again published a few years ago from our group, whereby we were looking at variability within diet and across cows in terms of, say, production variability related to this behavior of sorting. And I actually have the graphs kind of flipped around, but in the case of this study, what we did was we predicted the amount of milk fat and milk protein based on the amount of sorting cows were doing of long dietary particles. And so taking a TMR, total mixed ration, and shaking it out, and the stuff that sits on the top screen of the shaker box, we call the long particles. And basically what we do is we calculate then whether or not the cows are consuming the amount predicted as, or the actual amount versus the predicted amount. And so 100% would basically mean they're eating exactly as predicted, or anything under 100% meaning they're consuming less than predicted. And so for these long particles, long fibrous forage particles, they typically sort against those. And what we see here is this negative association, whereby basically those cows that do a lot more sorting against those long particles tend to have lesser milk fat as well as milk protein. And again, not the first time that this is being demonstrated, but just highlighting that, again, these are all cows eating same amount of feed on a dry matter intake level basis. It's the same diet, same production level approximately, but we see this huge spread in these milk components. And again, there's going to be obviously some genetic components to that, et cetera, but as you can see here, a significant proportion of that variation is also explained by the amount of sorting that those cows are doing of their diet. And again, that's explained by the fact that if those cows are not consuming that diet as it's put in front of them, then we're not going to get the production responses that we would expect from those cows. Another kind of example of that, just out of interest, was again a study published a couple of years ago from a former grad student, Rachel Kuhn, when she was doing her master's with us. Rachel did a project looking at feeding cows in early lactation one of two different types of diets, varying in physical effectiveness of the fiber in those diets. But what Rachel did in kind of a post analysis was to look at how production differences varied in those cows in early lactation based on their risk of ruminal acidosis. And so she classified cows as being either high or low risk of ruminal acidosis based on kind of area under the curve, amount of time and duration, or degree of decreased pH below a pH of 5.8 in the rumen. And what Rachel observed was that, interestingly, in the first 3 weeks of lactation, between these cows that we observed to be what we called low risk acidosis versus high risk, was that their level of feed intake was actually similar. Their milk yield tended to be a little less in those high risk cows. The milk fat content numerically was lesser, but statistically not, and there's quite a bit of variation, particularly in early lactation, so probably why we could not pick up any statistical difference there. But putting those two together, when we looked at fat-corrected milk, we did observe close to about a 15% difference in fat-corrected milk between those cows that were low risk and high risk. And again, I make this point because the only thing at the end of the day that we observed difference between these high risk and low risk cows was actually the amount of sorting that these cows were doing of their diets. The amount of dry matter consumed was the same, as you see in the top left there. And so just the composition of the diet based on the behavior of the cows can have a significant impact on the productive outcomes that we observe in these cows. And then the last kind of example or consideration when it comes to why we're interested in this topic area in general is the fact that feeding behavior may not only dictate outcomes that we see in cows, but cows may actually change their feeding behavior in response to physiological conditions. And we've demonstrated this in a variety of different studies over the years. This is actually an older study already from 2014. This was done actually with beef cattle in collaboration with Dr. Greg Penner out of Saskatchewan, whereby we were doing an acidosis challenge, rumen acidosis challenge on beef cattle fed a feedlot diet. And what we observed in the case of that study was that those cows or cattle, sorry, that experienced the greatest kind of change in duration of low pH. So in this case, rumen pH less than 5.5. So those cows that say increased by 10 hours or more per day from kind of baseline to post-ruminal acidosis challenge, we also at the same time observed an association with their change in sorting behavior. So whereby those cows sorted more for the long ration particles. And again, this is a feedlot diet with actually a very small proportion of forage in it. But even within that, those cows demonstrated, specifically those cows that experienced the greatest amount or greatest change kind of in their rumen pH, demonstrated a greater change to select for long forage particles, and fitting with other data that we have from our group as well as others suggesting that under these low pH conditions, the cows will actually change their selection pattern, their eating pattern to try to consume, say, more physically effective fiber to attenuate, reduce, lessen the effect of that low rumen pH. And what's interesting is that not only do we see that kind of change in eating behavior, particularly diet selection behavior in relation to maybe the rumen environment, we've also demonstrated that in relation to other kind of physiological status. And so this is a more recent study, again, from our group from Sydney Moore, who was a master's student with us a few years ago, who's currently doing her PhD at UBC. Sydney did a trial whereby we actually took high production cows in early lactation and induced a dietary, basically a dietary induced negative energy balance in those cows by switching those cows from their high production ration to a severely kind of diluted down diet. And what we observed was a transient negative energy balance, whereby cows, we observed a spike in NEFA, so a mobilization of fatty acids in those cows, and then at the same time observed how the cows changed their selection behavior, their feeding behavior, and in particular their diet selection behavior. And what you see here on the left side is the sorting of the long ration particles, so basically sorting of fiber, you could think of, and then on the right, the sorting of short particles, so the components of the diet, which would be more highly fermentable, right? So of highly fermentable carbohydrates. And so what you see is that in general, all the animals are sorting against the longer particles and for the shorter particles. We had two different types of diets. So basically one diet that was more easily sorted, that's the blue diamonds here. And the long is basically long straw versus short straw. But regardless of the diet, what we observed was that those animals that experienced the greater degree of fat mobilization or indication at least of some negative energy balance, those cows selected more against those long particles and more for those shorter particles. So again, indicating that those animals will change their eating behavior based on that physiological state. So that's in a nutshell, I guess, areas that we've been interested in from kind of a why perspective, and then obviously there's practical things that come out of that from a feeding management standpoint and how we manage diets, and again, lots of other work that we've done in that area, both past and present. But I thought for today, what I'd like to chat about for the rest of the presentation here is some areas that we're currently kind of exploring in our lab, and some studies that are recent and kind of ongoing. And the two kind of larger areas that I'll describe are how behavior comes into play when it comes to automated milking and our ability to, say, apply precision feeding within those systems, as well as, and actually related to that, the interaction of dairy cow personality with nutrition, health, and welfare. And I'll define that in a few moments for you as well. But starting with automated or robotic milking and precision feeding, one of the reasons we've gotten interested in this area, obviously, is that we see a large amount of interest and adoption of robotic milking in the dairy industry, particularly here in Canada over the last 5 to 10 years. And there's a lot of positives that come with that technology, one of which is a greater ability to manage cows at the individual level, and not just manage them overall, but even the feed consumption of those cows at an individual basis, whereby cows will still consume the majority of their feed from a common feed bunk with the rest of the herd, but we do tend to feed some level of feed within the robot, largely to actually just attract the cow to the robot. But at the same time, we can vary the amount of that feed based on the nutritional needs of that animal. And so we do have some opportunity there to potentially target and be precise in terms of the individual needs of that animal. Now, where there is opportunity there's also a challenge. And so one of the questions that we are looking at in a variety of different studies is how are we able to accurately do that, particularly at different time points throughout lactation. And I like to actually use the data that you see here to kind of highlight this. It looks like a bit of a scatter plot, but this is actual data points that come from our robotic milker at the Elora research barn, so our University of Guelph research barn. And so what we did was we pulled robot data from about a year and a half worth of cows that were kept on the robot at our research farm, and plotted out across days and milk of those cows the amount of feed that those cows were being offered within the robot. And what you can see is obviously quite a bit of variation there within kind of stage of lactation in terms of how much feed cows are getting individually at the robot. Now, some of you might look at this kind of optimistically and think, okay, this looks really good because we're actually able to, again, capture variation between cows in terms of maybe their production requirements and feed them individually. And that would be great if that was the case. But what I should point out is that actually within our herd in this time period, say from pretty well early lactation from about 3 weeks in milk to 120 days in milk, all of these cows actually had an allotment of close to about 6 kilograms of feed in the robot per day. And so now if we look at it with that in mind, we realize that while there are some cows in and around that amount, there's a whole bunch of cows that did not get the prescribed kind of level of feed that we had targeted for those animals within that robot during that time period. And there's probably a whole bunch of factors that may contribute to that, including just the individual visit frequency of those cows to the robot, the level of feed that they might be consuming at the feed bunk, influencing their desire to go to the robot and consume feed there, etc. And so part of our research program right now is trying to really elucidate that, to trying to figure out what factors might be contributing to some of that variation. And I'll come to that in a couple moments. One specific area that we're looking at is this early lactation period, whereby, again, when we get cows onto the robot in early lactation, we do potentially have some challenges in terms of getting enough nutrients into those animals. And we have some proof, I guess you could say, from a field study that we did already a few years back, whereby we demonstrated that the risk of ketosis, subclinical ketosis, a negative energy balance, was actually higher in cows that were producing more milk per kilogram of supplement they were receiving in the robot. And again, you could look at that and think, well, that's efficient, but it's not really efficiency. It's the fact that these cows were probably not consuming enough nutrients, particularly in the first 2 weeks of lactation, to meet the demands that they had. And some farms are often hesitant to, say, put too much grain into those cows, particularly in the first week or two of lactation, on risk of things like acidosis, and so might hold back a bit. But what we're seeing is that holding those cows back from an energy standpoint might actually be having a greater negative impact on those animals in terms of their overall energy balance. And so what we've been looking at or are interested in is trying to find ways of maybe mitigating that risk of negative energy balance, particularly through use of alternative feeds that, say, non-starch based energy sources that are going to be a little bit better, say, on the rumen environment of those cows, and potentially mitigate that negative effect there. And so a couple studies we've done. This was one, again, done by former student Sydney Moore, whereby we've fed a liquid feed actually in the robot, a molasses-based liquid feed, basically adding it in, dribbling it in with the grain that the cows were getting in the robot, and looked at how that affected the energy balance of those cows. While we did see some positive effect there, we saw a reduction in repeat kind of incidences of subclinical ketosis diagnoses. We still saw an overall huge level of subclinical ketosis. And this was a study of 400 cows in 5 commercial robot farms. And so still pointing to a challenge there in terms of the energy balance of these cows. We have since followed that up with some work trying to look at other alternative energy sources that could be used to, say, improve metabolic status and productivity of robot milk cows. And one of our current projects right now actually is one being led by current PhD student Brandon Van Soest. Brandon came to us from Michigan State about 1.5 years ago, and he's been working on robot-related projects. And this one specifically, we're doing at Alora, looking at pre- and post-partum supplementation of cows with a dry food-grade glycerol product, and looking at whether or not that has an impact on metabolic status and productivity. And so looking at obviously feed intake and sorting, blood metabolites, specifically things related to energy balance and inflammation in and around calving, rumination, standing and lying behavior, rumen pH profiles of those cows, which again, just novel from the fact that we haven't really seen any studies characterizing even the rumen dynamics of cows in early lactation kept on robots. Body weight, body condition score, milking activity, milk production, and components. And so Brandon's about 3/4 of the way through the project, should be done hopefully just past Christmas time this year, and we'll hopefully hear from him, and hopefully next year, if and when our group gets to present, then we'll hopefully have Brandon here to present some of the results of that project. And the idea behind this project is to take this again, once we're done this actually, is to take it out to commercial farms and actually look at supplementation of cows within robots within commercial farms to determine how well the glycerol supplementation works in that scenario. Another area that we've focused on kind of from this precision feeding approach is not only kind of the accuracy of the diet, but also the precision in terms of day to day. And this is a recognized problem particularly with feeding cows in robots, in that what our data and others has demonstrated is that we end up with more day-to-day variation in what cows consume as we try to target, say, more feed to those cows in a robotic scenario. And so this is some data from current PhD student Anna Schwenke from a study she did a couple years ago already, whereby we were investigating how 2 different levels of feed that we provide in the robot interact with PMR consumption and total dry matter intake of those cows. And what Anna observed, which is consistent with some other recent studies, is that the day-to-day variation, in this case measured as a standard deviation in intake across time, was higher in those cows fed the LPMR, which is basically a lower energy PMR, and a higher amount of feed in the robot. And while statistically there was no difference in the day-to-day variation in total amount of PMR consumed and the total dry matter intake, because of this variation, even though the total intake variation day to day was not different, the composition of that diet consumed would be different. And so that does pose a challenge for us in terms of trying to figure out why that's occurring and what factors might be contributing to that variability. And so Anna's actually looking at that in terms of in follow-up studies in her PhD research now, looking at what might be contributing to that variability, specifically looking at cow personality. And some of you might be taken back by that a little bit and think, oh, well, what is cow personality and what does that really mean? But really, from a simple definition standpoint, when we think about personality, just like you and I, our personalities, the classic kind of definition is individual differences in behavior that are consistent across time and situations. And so we know that, again, when we study cow behavior, that we do see, even though in general you'll see these certain patterns exist, individual animals will have very distinct kind of behavioral repertoires. And we can actually use various tests to actually classify those behavioral repertoires. And those behaviors then, that individuality in behavior, might actually have impacts then on how cows, say, respond in terms of these different situations. And particularly our interest is on the robotic milking side of things. And so one study that Anna's done recently in the last couple of years has looked at personality traits of cows milked in a robot in an AMS, and then determined how those personality traits really affect the response of those cows to different amounts of feed that we provide to them in the robot. So she varied the amount of feed provided to them, either 6 or 3 kilograms per day of concentrate in that robot. And the way that Anna assesses personality of these cows is to actually expose them to what we call a novel arena test, a novel object test, and a novel person test. So basically taking the cow, putting her in an area that she's not that familiar with, basically monitoring her behavior response, then placing a novel object in that area, looking at how the cow responds to that, and then finally putting a novel person who the cow is not familiar with, and then again monitoring or capturing the behavior of the cow and how she responds to that person. And then taking the collective behaviors that are recorded of those animals, actually subjecting those to a principal components analysis, a PCA analysis, to actually group the like behaviors. And what you see when you do that is you see kind of like behaviors cluster together. And then based on those behaviors that we're observing, we actually kind of ascribe, so there's a bit of an art form there, but ascribe different personality kind of profiles to those. And so based on, in the case of this study from Anna, on the novel arena, novel object test, she was able to describe personalities of being more active, more social, or more alert, curious. And then specifically to the novel person, cows that were either more active and vocal, as well as cows that were more fearful. And so you can imagine fearful would be basically cows that, either didn't approach or ran away from the novel person rather than approaching or being more active and curious in that situation. And so what Anna's demonstrated in this work is that those profiles then do correlate with the feeding or interact at least with the feeding approaches that we subjected those cows to in the robot. And so, for example, cows who were less active had greater day-to-day variation in their concentrate allowance at the robot when they're fed a higher level. And so that's this graph here on the top left here, where those cows that were less active, we actually see more day-to-day variation in terms of how much concentrate they're getting in the robot, versus those cows, say, on a lower concentration where there's really, it's almost flat, where we don't see that variability. And then same thing goes for the just total amount of concentrate cows were receiving in the robot. Largely those cows who were fed a higher level in the robot, their personality profiles, so cows that are either more alert, curious, less active, and more fearful, all had correlations of that behavior with the amount of concentrate that they consumed in the robot. So here, these cows that were, say, more alert, curious, had lesser concentrate delivered to them when they're on the high level of concentrate allowance. Same thing here for those that are less active, vocal, and then those cows particularly that were more fearful of the human, having less concentrate delivered to them. And so some really neat kind of associations there between basically how the individual cow's behavior profile influences how well we can kind of target nutrient consumption or nutrient intakes in those animals. And just a follow-up study to that, Anna's actually then taken that to look at how cows personality then affects their adaptability to an automated milking system for those cows that have never been, say, on a robot before. And so what she's done is actually looked at how boldness, which again correlates with probably the dominance of those animals within the pen, and their ability to, say, displace other animals from the feed bunk and from other resources, how that then influences their adaptability, say, to a robot. And just in the sake of time, I want to just jump to these results to show that basically those cows that were classified as being more bold in their behavior really showed a better adaptation to the AMS. So those cows had fewer days, or tended to have fewer days till their first voluntary milking visit. They had more voluntary milking visits overall and had a greater milking frequency post kind of adaptation to that milking robot. And so definitely the behavioral profile of these animals potentially has a negative, or sorry, a negative, has an association with their ability or their willingness at least to use that milking robot, and will then affect our ability to feed those cows individually in those systems as well. And then I'm just going to skip quickly just to give you one more example of a current project, just because I think there's probably a number of you which will find this quite interesting. We're looking at other areas where personality may have influences in dairy cattle, and the last project I want to highlight is MSc work of current student Brooke McNeil. She's actually working on this project in collaboration with Dr. Mike Steele's lab, and specifically with PhD student Lautaro Cagnano. And what Brooke's particularly interested in is how calf personality associates with gut microbiota, gut health, as well as the coping ability of calves at weaning to that stressful event specifically. And so what Brooke's interested in is trying to determine how basically the gut health of the animal, as influenced by a probiotic supplementation, influences not only the calves' coping ability at weaning, so basically how, from a behavioral standpoint and a state standpoint, affects their weaning ability, but how the personality of the calf then influences that coping ability. And then finally, and which would be very novel if we determine those associations, if we see any associations between the calf personality and the gut microbiota and gut health of those animals, and then specifically how that then too influences the response of those animals to a stressful situation. And so large project ongoing currently at the Ponsonby Research Station. Lots of tests, lots of different things from physiological to behavioral measures occurring, and don't have time to go through these all right now, but we're going to be looking at a whole host of different behavioral outcomes in these animals, as well as physiological outcomes to address those objectives. I just wanted to show very quickly just how we look at this. Again, I didn't show you videos of the cows, but in this case here, hopefully you can see here on the top left, this is a calf that's placed in a novel arena. And so again, we look at, from a personality standpoint, how that calf responds to that environment, and then how that calf responds to, say, a novel object, playing with that there, as you can see. And in the case of this study, we're not actually using a novel person, we're actually using a fear test, we're using a startle test. And what we actually do is attract the calf to the side of the pen with a milk reward, but as that calf comes by, you can't see the person behind here, but the calf will actually get squirted with a water gun right there, and you can see the calf react there. And so we'll actually measure the behavior response of that animal to that spray and quantify that, and use this information then to again come up with behavioral profile, personality profile of these animals, and then hopefully be able to correlate that back to their gut health and coping abilities. So concluding thoughts, it's not only important what cows eat, but how they eat it. Other things I hope to kind of convey as part of our research program is that there are other aspects of behavior, like the behavioral profile, the personality profile, that's also going to influence the success of nutritional programs for cattle. And that behavior then may reflect both the nutritional and health status of cattle, not only influence it, but also be a reflection of that. And again, these are areas that we continue to work on. So with that, thanks to my group, the Cow Crew, and here's just a array of the various current members of our group. And so thanks to them for much of the research I presented here today. And thanks to various funders of this work that I presented here. And hopefully we have time for some questions if people are still attentive and with us.
Wonderful. Thanks so much, Trevor. Really nice job. Any questions? You guys can write them in the comment section in the chat or speak up. Georgia, I think you put your hand up. Go for it.
Hey, Trevor, that was so interesting. And I kept finding myself wondering, especially in the first half, whether you ever look at stereotypic behavior, because I'm sure some of your cows will be spending time twirling their tongues and repeatedly licking at things. And if they're like horses, those oral movements will be a reaction to low gastrointestinal pH, and also potentially a way to cope with that by generating saliva. So I'm wondering if that could explain some of the variance in your earlier regression plots.
Good question. We've only seen that, really a lot of that in earlier work, again, stuff that we did over 10 years ago in Kempville, where we're looking at limit feeding of, say, dairy heifers, where, again, we're inducing feeding motivation in those animals, and then we saw that redirected behavior. We've never really seen it in response to, say, acidosis or anything like that in adult animals. Again, the problem with that behavior, and you're, I think, well aware of that too, it seems to be fairly individualistic as well, but then also it does get learned from one animal to the other without even the necessarily stimuli that induces it. And so that's something we observed too, that we had limit-fed animals and full-fed animals, and the limit-fed animals all started tongue rolling. And then a week later, all the full-fed animals were doing it too. So they actually were like looking at their neighbors going, oh, that looks like fun, I'm going to do that too, kind of thing. So,
okay, yeah.
That's
fascinating, because the social learning thing seems largely a myth, but it sounds like you've got good evidence, actually.
Yeah.
So that's cool.
Good anecdotal evidence. Yeah.
Thanks. Great.
Nice. Any other questions? Ming, I know you had your hand up earlier, but I think it went down. I'm not sure if you're still there. Any other questions?
Quiet group today.
Hi, Trevor. Do you think this personality is something that is genetic related? So my question is, like, thinking about the industry right now, do you think we should select cows who had better behaviors to adapt to situations, to the robotic milk system, or to a better transition period, or you should have like a different approach according to personality to improve this?
Yeah, no, it's a great question. And that's part of the reason why we're kind of going down this path is, and we've had this question before, I don't necessarily see us profiling cows and using that information for kind of day-to-day management of them. But where I do see this as potential is, right, if we can identify some of these associations, can we then, use that from, say, a genetics selection standpoint, right, to identify maybe populations of cows that may be more adaptable in their behavior, or have behavioral traits that are better for certain situations. We do know that some of that, like temperament, has been one that's been long used in selection indices. And there's more to behavioral profile than just temperament. We know that, right? So there's other things at play there. And so I think there is a huge component of nurture to this as well, right? And so that behavioral profile does change a little bit, even though they're supposed to be consistent over time. Major physiological events will kind of influence it. So work from other groups has shown that, like, even weaning in calves and puberty in calves will actually change their behavioral profile, right? And the consistency of their behavioral traits. And so we probably have to have a combination of those two things to kind of come into play there, right? Like we can use selection to some point, but then at the same point we still, like for nature, but then the nurture part still has to be there as well to kind of promote those, let's say, good behavior patterns that we want out of those animals.
Thank you.
Any other questions for Trevor? Certainly in the first half of your talk, Trevor, it's interesting from a precision feeding perspective as well too. And for us in nutrition modeling, you know, we always end up with all of this variation that we can't explain for. So the work on the individual feed sorting and stuff is really interesting as well in terms of, you know, how do we measure this, you know, in the future and be able to incorporate that information into these precision feeding systems.
Well, and that's, yeah, and I think that's the challenge, right? Is like we see that variation, like we know there's going to be, yeah, between animal variation just in terms of physiology, but then, yeah, beyond that, right, we can be as consistent as we want with what we're providing to that animal or on target, so to speak, but the animal's behavior still screws things up, right, so to speak. So
yeah, exactly.
Yeah.
Yeah. Okay. One more question from Marcos. Go for it.
Oops. Hi, Trevor. I just have a comment. For example, when we talk about the sorting for the top screen, like when cows have acidosis, they seem to select for the top screen. I think I don't know, it's a cause and effect thing. For example, I think the interpretation is when they're selecting for that, they get acidosis, but you could think if they have acidosis, they go there to solve the problem.
Yeah.
So don't you think a little sorting in favor of the top screen may be beneficial to those cows as far as it doesn't induce sorting against NDF? Like you could measure the sorting for NDF. So they're trying to solve the problem, so give the solution. So kind of.
Yeah, and I think you're dead on, and we've talked about this before, right? That, and I could have showed other data we have, right? So we have other data to show, yeah, if you allow the cows to sort too much, they can actually end up with acidosis. And then we have the other data to suggest that when we induce particularly a more significant bout of acidosis or ruminant acidosis, then they'll change their behavior to select for physically effective fiber, basically. I'm still torn whether or not we should just provide a diet that's not sortable that meets their physically effective fiber needs, and so we're not going to see acidosis anyways. So that might be the best approach. Or do we just give them another outlet, like some farms from a practical standpoint just have a tub of sodium bicarb sitting there, and if they see the cows go to the bicarb, they detect maybe an issue with the rumen in those animals, right? And that might be the indicator that there's something off in the diet. But the whole idea of self-medication, whether or not we should be providing those animals, I think is interesting. I'm kind of torn there though. So.
Okay.
Good. Thanks, Marcos.
Thanks.
Great. Well, we're a little bit over time, so we'll probably wrap it up there. Thank you so much, Trevor, for a really interesting talk. Really appreciate it. And we'll leave it there. We do have another CNM seminar coming up in December, so it's going to be the Mike Steele Lab up next. So stay tuned for that meeting invite in your inboxes soon. And otherwise, thank you so much, Trevor, and have a great day, everyone. Thanks for coming.
Jen. Take care.